This is Epithelium, Junctions, and Transport. This tutorial is designed to answer some basic questions about how epithelial cells are designed to perform their basic functions of protection, secretion, and absorption. Today we'll explore how the structure of epithelial cells informs those functions. I'm Dr. Catherine Moore, the Histology Wizard. Now, what properties do epithelial cells have to perform their different functions? Now here I'm showing a generic cartoon of a couple of epithelial cells, and I'm going to use this diagram today to discuss different characteristics. Let's take skin for example. This tissue is designed to line the body surfaces, so it needs to be leak-proof. Recall that epithelia are continuous sheets of cells, and that skin is subject to a lot of friction and mechanical forces, so cells need to strongly attach to one another to be able to communicate with one another, and they also need to strongly attach to the underlying basement membrane and connective tissue. To solve these problems, epithelial cells contain specialized membrane-associated structures, and these provide adhesion and communication between cells. Now, this cartoon summarizes those different types of membrane-associated structures, and we're going to now walk through those one by one. What else do cells have to do? Well, cells have to be able to transport substances into the cell and to remove substances from blood and tissue to the outside of the body. So these cells also have to have specialized structures to control absorption and secretion of ions, proteins, and water, etc. Also recall that epithelial cells are polarized. So they have an apical side, which faces the free surface, and a basal side that's closer to the underlying connective tissue. Now on the apical side, the cells can have modifications, and these are specialized structures that can add function. So here we see mobile cilia, and they help with the movement of substances, as we see in the respiratory epithelium. Next we see microvilli, and these are sort of bristles that help increase the surface area as needed in the cells of the duodenum, where nutrient absorption is high. And here's a comparison of cilia versus microvilli. Now that you're oriented, we're going to walk through those membrane structures one by one, and we'll start with the junctions that are located on the apical surfaces. And as we go, try to predict how defects in these structures might lead to changes in the ability of epithelial cells to perform different functions, because these defects can lead to disease. So here we have a cartoon of an epithelial cell sheet, and I've labeled the apical and the basolateral membranes. Now we're going to zoom in on two cells and talk about the junctions, starting close to the apical surface. The first intercellular junction we'll talk about is the tight or occluding junction. Sometimes you'll see this called zona or zonula occludens. Now, one of the things you have to get used to in histology and anatomy is that the structures often have many different names. The problem is some of these names are historical, they're named by anatomists, sometimes for anatomists, and sometimes they refer to the terms that a pathologist might use. But don't worry, you'll soon get used to this. And in fact, the names can sometimes provide clues as to the function of the junction. Zonula means small belt in Latin, and that indicates that these junctions can form a band completely encircling the cells, in the same way that you put a belt around your waist. So what do tight junctions do? They form a continuous intracellular barrier between adjacent cells. They serve to prevent passive flow of material between the cells, or so-called paracellular transport. You can think about tight junctions like a seal on your water bottle. Their job is to prevent leaks. In other words, a tight junction ensures that molecules that cross an epithelium have to do this by going through the cells, and they can't sneak or leak between them. And we call that transcellular transport. Tight junctions are like seals on water bottles. Now, an epithelium that needs to be leak-proof, such as in the bladder, will generally contain more tight junctions than cells that require permeability to function, such as cells in the kidney tubules. And tight junctions also help set up the apical and basal compartments. Now, for each one of these junctions I'm talking about today, they have very specific proteins that are associated with them. And tight junctions have occludins, claudins, and zooproteins. What happens when these junctions don't function properly? Like a faulty seal on a water bottle, they leak. And mutations or disruptions of these proteins can lead to disease. For example, food poisoning can be caused by bacteria that targets these tight junction proteins, and that interrupts the formation of the junctions, and that causes loss of tissue fluid into the lumen of the gut, and that results in diarrhea. Moving basally, our next junctions are the adherence junctions, or zonula adherens. They're found close to tight junctions, and like tight junctions, they form a continuous band or belt around the cell. These serve to stabilize and hold cells together, similar to the plastic rings that hold a six-pack of Diet Pepsi or Diet Coke together. And loss of cadherins, which are the protein members that are associated with adherence junctions, can lead to loss of cell-to-cell adhesion. So again, in adherence junctions, those main protein components are members of cadherin families, and they attach to actin filaments of the cytoskeleton of the cells that are side by side, holding them together. Our next junction, a little more basally, is another anchoring junction called the desmosome, or macula adherens. Now, desmosomes are structures at the surface of one cell that are matched with identical structures on the adjacent cell surface. Unlike the tight junction and adherence junctions that we just talked about, desmosomes don't really encircle the cell like a belt, but they act more like single spot welds, although they are all over the cell. I like to think about desmosomes as Velcro. Specifically, I think about them as those industrial strength Velcro dots. Like Velcro, desmosomes interconnect for a super firm hold. And when these get pulled apart, this results in blistering disorders. Cells now in that epithelial sheet will come apart. Cell adhesion by desmosomes is mediated by larger members of the cadherin family. These are called desmoglines and desmocollins. There are also catenin-like proteins called placoglobins and desmoplakins, and these link to intermediate filaments rather than actins. And they contain different keratins. Let's pause just for a minute and summarize. On the left in this cartoon, you see those three intercellular junctions: the tight junctions, the adherence junctions, and the desmosomes. On the right, you see what those junctions look like under the electron microscope. And what I want you to note particularly is that kind of bristly bottle brush appearance of the desmosomes. And those bristles sort of coming out from the center are those intermediate filaments that really hold those cells together. So now I've shown you how cells can attach to one another and how they can prevent leaks between the cells. But we also know that the epithelium has to attach to the underlying connective tissue. On the basal epithelial surface then, cells attach to the basal lamina by another set of anchoring junctions called hemidesmosomes. And you can see these kind of look like half a desmosome. But the proteins in desmosomes are called integrins, and they directly bind to laminins and to intermediate filaments in the basal lamina. These are keratins, but they're different from those in desmosomes. Ultrastructurally, as I mentioned, these junctions resemble a half desmosome. So you can see sort of the bristly side here. And like a desmosome, blistering diseases can be caused by mutations in keratins or integrins of the hemidesmosome. There's a related basal anchoring junction called the focal adhesion junction, and this also has integrins, but they link directly to actin filaments rather than keratin intermediate filaments. Finally, remember that cells not only have to adhere to one another, they have to be able to communicate. And gap junctions are a type of communications junction. So rather than adhering or occluding, they mediate that intracellular communication. Now these junctions are actually complexes of transmembrane proteins that form kind of patches in the plasma membrane. And you can see them here on the left in this electron micrograph just as little circles. And in the next cartoon as kind of those little passageways, or they almost look like hollow plugs between the different cells. Now there are connexin proteins that form channels that permit intracellular exchange of small molecules and ions. And this exchange allows cells to act in a coordinated manner. So the best example of this is heart contraction. And mutations in connexins can lead to all sorts of abnormalities. This can include deafness or heart arrhythmias such as atrial fibrillation. So let's review. In order from the most apical structures, we have tight junctions that help control water transport and leakiness of the cells, adherens junctions and desmosomes to help epithelial cells adhere to one another, hemidesmosomes to help epithelial cells adhere to the basement membrane, and gap junctions that allow cell-to-cell communications. So the next big thing that cells need to be able to do is to control substances in and out, including ion and water transport in either direction. And cells do this in two typical ways. The first is called transcellular transport. So in this case, substances actually pass through the cell membrane. Second is called paracellular transport, where mostly water and ions pass between the cells. And paracellular transport is tightly controlled, in part by tight junctions, which we just talked about. Most cells have both kinds of transport, but for example, capillaries in the blood-brain barrier don't have paracellular transport. And this actually makes a lot of sense since the passage of substances into the brain needs to be tightly controlled. Now, control of ion and water movements is a critical function of epithelial tissues, and misregulation of this process can have very serious consequences. And there are a number of different ways that molecules pass membranes. So substances such as oxygen, carbon dioxide, and some hormones can pass membranes via simple diffusion. They're kind of like ghosts, as my colleague will say. Some can pass through different channels, such as water channels, gated ion channels. Some pass through antiporters or symporters. And some transport can require energy. Epithelial tissues need to be able to actively transport ions against concentration and electrical gradients. And so a good example is this sodium-potassium ATPase or sodium-potassium pump. And that allows cells to maintain a required low intracellular sodium concentration and have a large excess of potassium ions on the inside of the cell, aka the salty banana. Now, what about water? How does it cross the membrane? Well, as I implied before, it does this two ways. First, it can move transcellularly through the membrane via channels called aquaporins, seen here in blue. If aquaporins are inserted into the membrane, water will flow through. This happens via osmosis, and the direction is determined by the relative solute concentration. And water can also move paracellularly. Either way, ion channels are important in determining the direction of water flow. Now, we will continue to examine transport in much more detail as we discuss different organ systems, but for now I want to look at two simplified diagrams of an intestinal epithelial cell. I'm going to show some of the important transporters, channels, and junctions involved in ion and water transport. Now note, I'm not going to show all the channels. I'm just going to illustrate some general principles. So let's walk through it together. First, we will talk about absorption, or transport from the lumen outside the cell, across the apical surface, and eventually into the blood. Now remember again that apical is going to be up in this cartoon, and that the cell is a salty banana, with low intracellular sodium and high potassium. So absorption is mainly driven by active sodium absorption, and this can happen through specific ion channels on the apical surface. ENaC is the one that I've shown here in red, and it controls influx of sodium. Now to pump the sodium out of the cell, because again, we want to keep low intracellular sodium concentration, there's that sodium-potassium ATPase, or pump. And that active transport process involves the hydrolysis of ATP to provide energy that's going to drive sodium out and potassium into the cell. And there's also a potassium channel that will help drive potassium out to maintain this balance. Now together, these channels control the flow of sodium into the cell and into the interstitial tissue and eventually into the blood. And they also create an electrochemical driving force and this helps bring in chloride paracellularly. Now, in addition, because water always follows sodium concentration, water is going to flow into the cell. And again, it's going to come through aquaporin channels, transcellularly, and it will also flow through the paracellular pathway. And remember, the point here is to maintain intracellular sodium low and intracellular potassium high. And changes in the balance can lead to dysfunction and disease. Now let's talk about secretion, or transport of ions and water from the blood and the interstitial tissue across the basolateral membrane into the lumen and in some cases outside the body. We're going to use the same basic diagram, but now I want you to look on the left where you can see the normal concentrations of sodium and potassium. Remember, that's salty banana. Secretion is driven for the most part by chloride secretion. So this time we're going to start on the basolateral membrane where there are several transporters that all function to help accumulate chloride inside the cell. And I've shown those in yellow and orange. And these include a sodium-potassium chloride cotransporter, and that aids in active transport of those ions across the plasma membrane. And this works in concert with the sodium-potassium pump we just talked about. And in epithelia, this NKCC channel is critical for chloride-driven secretion. And also we have some anion exchangers and a sodium bicarb exchanger that all help in chloride-driven secretion. On the apical membrane, there's a channel called the cystic fibrosis transmembrane conductance regulator, or CFTR for short, and I've shown it here in green. In addition, there are other chloride channels, also shown in light green, that help control the secretion of chloride out of the cell. In part, the CFTR channel also functions to control that ENaC sodium channel that I showed on the previous slide. So to sum it up, CFTR alters sodium transport transcellularly, and chloride secretion helps drive sodium movement across the epithelium paracellularly. And then the water will follow the sodium, again, through aquaporins and also the paracellular pathway. Now together, along with other channels and transporters, these transporters balance the processes of secretion and absorption in epithelial cells, and this teeter-totter can change according to the needs of the specific cell type. Now mutations in these transporters then causes misbalances, and that leads to disrupted ion and water transport and thus disease. So let's finish by taking a real-life example. So we're going to talk a little bit about cystic fibrosis. Mutations in the gene that encodes CFTR protein can lead to imbalances in the chloride and sodium ion movements, and as a result, changes in water transport. Now, the consequences of the mutation depend both on the type of mutation and the role of the CFTR channel in a specific organ. So we're going to look at the respiratory system. So here, mutations in CFTR can have drastic effects on health. This diagram shows a healthy epithelial cell in the lung. And for simplicity, I've just shown a chloride, sodium, and water channel on the apical surface. In this case, we have a normal balance of secretion, absorption, and water movement. And this results in a thin, watery mucus. And this is normal. It helps get rid of particulates and other things we don't want in the lung. And as this cartoon on the right shows, this results in healthy airways that have ability to conduct a lot of air into the lung for gas exchange. Now, in the case of defective CFTR protein, the amount of secreted chloride is reduced drastically. And in addition, since CFTR normally inhibits the ENaC sodium channel, the overall effect is that there's an increase in the amount of sodium transport into the cell. This disrupts the gradient. And because water will follow the sodium, this is going to decrease the amount of fluid in the lumen of the respiratory airways. And that leads to a thick, sticky mucus that traps more bacteria, increases obstruction and infections. It can't be moved out of the lungs. And we can see the end results of this lack of movement of the mucus diagrammed in the cartoon on the right, where the airway becomes blocked, and this in fact affects both conductance of air into the lungs and gas exchange. CFTR mutations in the gastrointestinal tract act in a similar way, such that intestinal secretions are abnormally thick and sticky, and this decreases gut motility that can cause constipation or even bowel obstructions. With this example, that's it for junctions and transport. Don't worry if you can't master all this material at once. We will be talking about specific structures and functions of different epithelia in various organs, so there'll be plenty of opportunities for reinforcement of major concepts and details. Thanks for stopping by.